Dr. Juarez is a lecturer in physical chemistry at the Faculty of Pharmacy at the University of Barcelona, and also a group leader of the Computational Molecular Design Lab. He obtained his PhD by the University of Barcelona in 2014 for modeling in drug design, and he then conducted postdoctoral studies at the University of Florida and University of Edinburgh. And at the moment, his research focuses on using molecular simulations in the field of computer-aided drug design, with particular interest on how computer-aided drug design can be useful to generate rational approaches for the design of small molecules that induce protein proximity. So I know Jordi's interest has been looking at molecular glues specifically, and this is something that is also very close to the theme of the webinar. So it's going to be very exciting to hear how you try to disentangle these interactions. Thank you so much, Jordi, and the floor is yours.
Share the screen.
Yeah, we can all see it properly.
Yeah, perfect. Right. So thank you, Radek, for the invitation and to give me the opportunity to explain you a little bit what we have been doing in my research group. I have to say that unfortunately, I don't work at IRB. I'm only work at the university, but we'll see in the future. But I would like to start with a little bit of personal story, because one thing, when I'm a trained pharmacist, and when I was doing the degree, the story of thalidomide was really shocking to me, like how that could happen at the time, even though the studies were conducted at the standards of the industry. It was very shocking, and it was also eye-opening learning, because what I realized is that actually what happened is that people knew what the thalidomide was doing, but they didn't understand what was going on, right? It took several decades before we were able to actually propose a mechanism of what was the molecular mechanism that was driving the different effects that were described for thalidomide. And I think that's actually, that contains cautionary tale for the field, in the sense that it's very important to bring rational approaches and to bring, when we deliver compounds into the clinic, it's very important to follow a rational, have a rational understanding of what's going on. And I think that sometimes in targeted protein degradation, that's not so evident. And I think that the call for more targeted, for more rational approaches in the field have been highlighted for several years now. And I also think that computational chemistry can have a role in helping expand these rational approaches. There is 4 specific areas where I think we can be useful, either identifying new TPD effectors and new degradable targets, generating predictive and reliable models, or developing integrated computational workflows. I think these all are areas where computer-aided drug design is can have a very important role. But today, and currently my focus is more on the more basic understanding, like trying to understand what are the physicochemical phenomenon that drives targeted protein degradation, specifically with molecular glues, right? And because I think that when you put the molecular glue mechanism in a physicochemical perspective, right? You can imagine that in the crowded space of a cell, you have at any given time a very high number of transient contacts, right? Like the proteins are touching each other, and these transient contacts are very difficult to observe by experimental techniques because they are high on energy. They are not going to be long-lived, right? But nevertheless, they exist, and you can see it because you have molecular glues that stabilize them, right? So at some point, these contacts are forming. These molecules are coming into contact somehow, and you can imagine this as a very free energy surface that is very rough with a lot of very shallow local minima. And then when you introduce the molecular glue, you get one minima that gets stabilized, right? And gets suddenly that ternary complex is observable by experimental techniques. And to me, this poses the very intriguing possibility of, okay, if we can understand how this process is happening, we can exploit it for designing new molecules and designing new strategies, right? And that's what we're trying to understand. And we started small, I would say, like with a very intriguing molecule that is the complex between Cereblon, CK1 alpha, that is stabilized by Lenalidomide. And the fact that I found this complex so intriguing is because if you look at the crystal structure, right, you can see that Lenalidomide sits in the binding site of Cereblon, right? And then you can see that most of the most important interactions are 3 hydrogen bonds that are established between the loop, this hairpin loop in CK1 alpha, and the tryptophan cage moiety of Cereblon. And then if you remove Lenalidomide for the picture, to me, there is not evident why these hydrogen bonds shouldn't be there. Right? You should be able to form the same interactions, regardless of whether Lenalidomide is there or not. Nevertheless, when you compare the stability of both complexes, you have 100-fold difference in the stability of the ternary versus the binary complex. So the way that we think about this is that you actually have a cooperative mechanism going on, right? So somehow there is something in this ternary complex that makes it more than the sum of its parts. And what we hypothesize is that whatever was going on had to do with the strength of these 3 hydrogen bonds that are at the interface between Cereblon and CK1 alpha. So we had or we developed a protocol that could allow us to computationally evaluate the strength of the individual hydrogen bonds, right? Using what we call stereomolecular dynamics, right? Basically, what stereomolecular dynamics is if you have one hydrogen bond interaction that you want to stretch, right? You can make the program to stretch it at constant velocity, right? If you were to do this at the infinitely slow speed, right, you will be able to recover the free energy or difference, right, between the free energy profile that is required to break this hydrogen bond. Right? However, you cannot, in practice, you cannot do things infinitely slow, and then you have to do it at a certain speed. When you do this same process at a certain speed, what happens is that you get a value that is different from the value that you will get when you do it infinitely slow, right? And then this distribution of values, according to the second law of thermodynamics, will follow a distribution that it will be such that the average of these values will be always equal to this work that you got, like this free energy, plus a certain amount of this dissipative work. And this value will always be bigger than the free energy required to break this interaction. So it's sort of a ceiling of what that free energy is, which is interesting, can be informative, right? But then in 1990, it was published this equality by Jarzynski, where he was able to relate a free energy value with out of equilibrium works, right? And that's very interesting for us because it means that if we manage to do this process a number of times, we can treat the data so we can recover the free energy from this distribution of works, right? So with this technique in place, we said to, okay, let's gonna take the 3 hydrogen bonds that are established at the Cereblon CK1a interface, and we're gonna break them in the presence and in the absence of lenalidomide. And what we saw was quite striking, because we saw quite a big difference in the strength of these hydrogen bonds when lenalidomide was present. Right? You can see here in the 3 panels, right? The line at the bottom, that is the one corresponding to the ternary complex, is always over the line at the top, right? Which is the corresponding to the binary complex. And that was really, really interesting, because it meant that really the lenalidomide was strengthening these hydrogen bonds, right? And we hypothesized that what was happening, right? Our hypothesis at this stage was that, okay, what this lenalidomide is doing is blocking the access of water molecules to these hydrogen bonds, right? It is a well-known concept that hydrogen bonds that are solvent exposed are very labile and easy to break, while hydrogen bonds that are buried in the structure of the protein are a lot harder, right? And the current theory is that this is because water can act as sort of a catalyst, right? It will replace one of the interacting groups, right, and make this hydrogen bond easier to break. So what we did in our simulations, we evaluated how accessible the hydrogen bonds, right, were when lenalidomide was present and when it was not. And then to do that, we use what we call radial distribution function, that basically counts like the density of water molecules that are around a certain point. And you can see in the panel that in all cases, right, but specifically in the 2 on the sides, when the ternary complex against the orange line, you have a big difference in how exposed these hydrogen bonds are. Right? So we said, okay, that's it. We got it, right? We know that if the RDF is low, that hydrogen bond will be hard. If the RDF is high, then that hydrogen bond won't be as difficult to break. So we went and we test whether different mutants that were described for CK1 alpha at the time, right, and their interaction with Cereblon, right? We went and looked at whether the hydrogen bonds were affected, right? And we could see that hydrogen mutants of CK1 alpha that decrease the stability of the ternary complex were also mutants that in greater or lesser measure were affecting the strength of at least one of the hydrogen bonds, and in some cases, the 3 of them. That was really exciting. But when we went and looked for the RDF, what we saw was a little bit less exciting, because the differences in RDF are not so evident. Right? You can see in these different panels that regardless of the effect that lenalidomide, that the mutant had on the hydrogen bonds, right? You have some cases where, for example, the right-hand panel here, you can see that, you can see that the 3 hydrogen bonds, or especially these 2, they are very decreased in strength with respect to the wild type CK1 alpha. That will be the gray line at the top. Right? And then when you compare the RDF, it's really similar to the one of the wild type. Right? So at this stage, we're a bit puzzled, because on one hand, it looked like it was that the preventing the access of water molecules was it makes sense that was preventing was preventing the rupture of these hydrogen bonds. But on the other side, when we look at the equilibrium structures of these hydrogen bonds, we were not able to see these differences in water access. So we went and we looked at the trajectories right? And what we did basically was to select manual check or many of them and compare when we had a trajectory that was high energy, high work, and trajectories that were low work. And what you will see now is a selection of representative trajectories, but they are not cherry picked. They are actually representative of what we were seeing. And for example, right, you have this breaking. You see this trajectory where we had a low work, right, value at the end, and you have a water molecule that was accessing. This is the same hydrogen bond, high work value, no water molecule. The same for the hydrogen bond in the middle, right? High work, right? Water molecule appears after, and then this hydrogen bond, low work. You have here the water molecule appearing, high work, no water molecule inside when this is breaking, right? And then in the mutant, well, you see that you have low work, a water molecule is appearing. So at this time, our explanation was like, okay, it's not a matter of what these systems, how the systems are behaving in the equilibrium structures, but it's a matter of what's going on while they are breaking. So even if your mutant affects the dynamic process of breaking, right, facilitating the entrance of water molecule at this point, you will also have a decrease in the stability of the hydrogen bonds. This is a very interesting theory, but it's also not provable directly because there is no experimental value that you can relate directly to the strength of a single interaction, right? You always change something else. So what we tried is, okay, can we use this as the combined stability of the 3 hydrogen bonds as a reporter of the stability of the whole ternary complex? And to do that, what we did was to convert the KDs into values of free energy of the stability. And then we saw that we got a very good correlation when we were comparing the total strength of the 3 hydrogen bonds with the stability of the ternary complex. And this was shocking because in principle there is no a priori reason why these two values should be correlated, right? As you know, free energy of binding is equilibrium quantity, and the way that we compute the energies of breaking the hydrogen bonds, it's an out of equilibrium process, right? So it shouldn't be correlated, but we saw that you could actually predict whether the ternary complex will be stable or not based on this combined strength. And our interpretation of this is that actually what we are observing is a characteristic of the mechanism of the way that Cereblon and CK1 alpha interact, right? So if you think about the mutants that we test of CK1 alpha, they are really unlikely to affect the K-on, right? Because they are on a side, they don't involve great conformational changes. But then if you make these hydrogen bonds harder to break, right, you will affect the K-off, right? So if you have if you are affecting the K-off, essentially you are affecting the KD. That's our interpretation, and that's what the simulations are telling us. Right? So this is our hypothesis of how lenalidomide is actually stabilizing Cereblon CK1 alpha contacts. Basically, when you have no lenalidomide in the interface, you have a relatively exposed protein-protein interface where water can enter and it can help break these hydrogen bonds and results in very labile interactions. Right? when you have lenalidomide, it will prevent the access of water molecules, right? And then you will have hard, robust interactions that are difficult to break, and that gives you a very stable ternary complex. And then if you have mutants that somehow allow better entrance of water molecules, right? Because you reduce the side chain of the size of a side chain, for example, those mutants will be also less stable in the ternary complex because you will have an exposed hydrogen bond. That was our hypothesis, and was was picked up by Derek Lowe, which made us very happy. It was quite a surprise, and I hope that just want to conclude with final remarks. Basically, I think that the affinity, or we hypothesize that the affinity of lenalidomide mediated Cereblon CK1 alpha complexes relies on the strength of the 3 hydrogen bonds, and that's the main contributor. This strength of hydrogen bonds can help predict the stability of ternary complexes, and we think that Cereblon CK1 alpha is proof of concept of a more general mechanism, right? And currently we are working in 2 ways. One, establishing how general this mechanism is, both inside the cerebellum space and outside the cerebellum space. And also we are working on trying to incorporate these measurements in prospective studies, right? At the moment we have, in order to do this, it takes, well, it took quite a lot of simulation time to do properly. And we are trying to find ways to speed up this process so we can link it into virtual screening cascades and help us to identify new clues. I would like to acknowledge the work of my tiny research group. Also the work of Marina Miñarro, which was the main contributor to the article, and also Andrea Bertran and Jorge Duro, who were helping her and me. I would like to acknowledge the collaboration with the groups of Professor Barril and Dr. Galdeano, the funding entities, and the Barcelona Supercomputer Center for providing the computational muscle that we needed in order to actually complete the calculations in a timely manner. I would also like to let you know that we are recruiting for a PDRA. If you are interested, get in touch, right? Feel free to send inquiries. And then with that, I will be very happy to take your questions.